Welcome to the Bio Insights, where science comes alive.
Today, we're diving straight into one of the absolute most revolutionary techniques in modern biology, the polymerase chain reaction, or, as we all know it, PCR. Let's get right into it. Before we jump into the actual mechanics, picture this. What would you do if you had only one tiny, totally degraded piece of DNA? Maybe it's a super rare sample from a lab disaster or a microscopic trace from a crime scene, but you desperately need millions of copies to actually run your research. You definitely can't just slide DNA into a regular office copy machine, right?
Well, the superhero solution to this massive problem arrived back in 1983. Fun fact from our sources, that's actually the exact same year India won its first Cricket World Cup. That year, a scientist named Kary Mullis invented PCR. It was a completely brilliant, game-changing method for cell-free DNA cloning. And honestly, it was a discovery so profound that it earned him the Nobel Prize just a decade later in 1993.
So, here is our roadmap. We're going to break this complex process down into 5 clear sections. First, PCR as a molecular photocopy machine. Second, the lab version of DNA replication. Third, the 3 main steps of PCR. Fourth, primer design rules. And finally, the math of PCR.
Starting off with Section 1, PCR, the molecular photocopy machine. You know, what's really fascinating here is how PCR isn't framed as some completely new, unimaginable biological concept engineered from scratch. It's actually just a clever laboratory mimic of what our own cells do every single day. The key word here is in vitro. Basically, this natural process of DNA replication is pulled completely out of the cell and performed in a highly controlled artificial environment, like a tiny test tube.
Moving right along to Section 2, the lab version of DNA replication.
This comparison brilliantly illustrates the core analogy of PCR. Think about it. To replicate DNA inside a living cell, biology relies on incredibly complex proteins. To unwind the DNA, nature uses the helicase enzyme. But in the lab, scientists completely bypass that complexity and simply use extreme heat. To start the copying process, nature uses the primase enzyme, but in the lab, we swap that out for artificially synthesized primers. And to actually build the new DNA strand, nature uses DNA polymerase III, while the lab uses a highly specialized alternative called Taq polymerase. It's biological engineering through absolute simplification. So, to run this molecular photocopier, you need a very specific recipe mixed into your PCR tube. You've got to have your template DNA, which is your master blueprint. You need your synthetic primers, both forward and reverse, to flag exactly where the copying should begin. Then you need Taq polymerase, acting as the microscopic builder, and a pool of DNTPs, which are really just your free A, T, G, and C nucleotides, acting as the literal building blocks. And finally, you stabilize the whole shebang with magnesium ions and a buffer solution. The absolute rock star of this mixture is, without a doubt, Taq polymerase. Look, if you tried to use human DNA polymerase in this machine, the heat would destroy it instantly. But Taq polymerase is a thermostable enzyme isolated from a bacteria called Thermus aquaticus. Because this specific bacteria thrives in naturally boiling hot springs, its enzymes have evolved to comfortably survive the intense, near-boiling heat of the PCR process without denaturing or falling apart. But hey, we also can't forget the unsung hero of this process, magnesium. It acts as a vital structural bridge in this microscopic world. See, the DNA phosphate backbone is negatively charged, and the active site of the polymerase enzyme is densely packed with negatively charged amino acids. Normally, they would repel each other aggressively, kind of like 2 identical ends of a magnet. But the positively charged magnesium sits right in the middle, creating a bridge that lets the polymerase physically interact with the DNA without pushing it away. Pretty neat, right? All right, now that we have our ingredients perfectly mixed, let's look at Section 3, the 3 main steps of PCR.
The thermocycler machine repeats 3 distinct, temperature-driven steps over and over again to build our DNA copies: denaturation, annealing, and extension. The entire process is literally just a highly calculated manipulation of temperature.
In the very first step, denaturation, the machine cranks the heat way up to between 94 and 95 degrees Celsius for about a minute. This extreme heat acts just like our artificial helicase. It literally forces the hydrogen bonds holding the DNA helix together to melt and break apart, cleanly separating our double-stranded DNA into 2 exposed single strands. This basically opens the book so we can actually read the genetic code. Next up, we quickly cool things down. In step 2, annealing, the temperature drops to somewhere between 55 and 65 degrees. This specific cooling zone is where our synthetic forward and reverse primers can successfully find and stick to their complementary target sequences on those single DNA strands. It's an incredibly precise operation, honestly. If the temperature is slightly too high, the primers won't stick at all. Too low, they might bind to the wrong random places, totally ruining our results.
Finally, in step 3, extension, we raise the temperature back up to exactly 72 degrees Celsius. This happens to be the absolute optimal working temperature for our hot spring superhero Taq polymerase. The enzyme locates those newly attached primers, grabs the free-floating dNTP building blocks, and rapidly cruises down the single strand. It extends it by matching up the complementary pairs until an entirely new, double-stranded DNA molecule is fully formed.
Let's move on to section 4. Primer design rules. The ideal length for a synthetic primer is strictly engineered to be between 15 to 30 base pairs. This range is absolutely crucial, because if a primer is too short, statistically speaking, it'll find matches randomly all over the vast DNA genome, giving you chaotic, non-specific copies. But if it's too long, it struggles to attach efficiently and bogs down the entire amplification process. A perfect primer also requires a super precise chemical balance, specifically containing between 40 to 60% guanine and cytosine, the GC content. Why? Well, G and C nucleotides form 3 strong hydrogen bonds with each other, whereas A and T only form 2. So hitting that 50-ish% sweet spot ensures the primer anchors firmly enough to the template to stay put, but not so strongly that it can't be easily separated in the very next heat cycle. To figure out the precise temperature where exactly half of your primer is bound to the DNA and half is separated, scientists use a really handy mathematical shortcut called the Wallace rule. By multiplying the number of G and C bases by 4 degrees and the A and T bases by 2 degrees, they can predict the perfect thermal sweet spot, known as the melting temperature or TM, based entirely on the strength of those hydrogen bonds we just talked about. And this brings us to a golden rule of primer design. The number 2. This number is incredibly important because the calculated melting temperature difference between your forward primer and your reverse primer must never, ever exceed 2 degrees Celsius. If one primer prefers 55 degrees and the other prefers 60, finding a single annealing temperature for the machine is just impossible. One primer will attach perfectly while the other completely fails, and your whole reaction will crash.
For our final section, let's look at section 5, the math of PCR.
Because every single newly minted copy of DNA instantly becomes a fresh template in the very next thermal cycle, the total amount of DNA grows at a staggering exponential rate of 2 to the power of n, with n simply being the number of cycles. If you run 30 cycles, one single piece of DNA can become over 1 billion copies in just a few hours. Now, because laboratory conditions and enzyme stamina aren't totally flawless, the real-world formula accounts for the fact that machine efficiency actually hovers between 60 to 90%.
So I want to leave you with this provocative thought to ponder. Taq polymerase is famous for being an absolute unit, surviving near boiling temperatures to build DNA at incredible speeds. However, as our sources point out, it entirely lacks 3' to 5' exonuclease activity. In plain English, it has zero proofreading ability. It literally cannot back up and fix a typo if it accidentally adds the wrong nucleotide. So if our superhero enzyme is essentially working blind and incredibly fast, what does that mean for the accuracy of our final billion copies? How do scientists trust the results if a single early typo gets amplified millions of times? Just a little something to think about the next time you hear about a PCR test. Thanks for joining me on this explainer as we decoded the microscopic world.